My name is Nicole Masters, agroecologist, systems thinker, author of the book For the Love of Soil. If you haven't got a copy, do grab one. It's also on Audible Books. With my company, Integrity Soils, and my trusty sidekick, Megan, wherever she is, she's American, they stand out. Yeah, back there. Very good. Thank you. We're with Integrity Soils. Before COVID, we were working directly with about 1.75 million acres consulting and coaching in North America and Australasia. COVID kind of changed things, and it's been great because we now work with organizations shaping what does it look like to truly regenerate with transparency and authenticity, and we work alongside 29 million acres right now. So if anyone says this isn't happening, we're at the tipping point in regions, so get excited. Now, I was asked to do an advanced class. Hold on to your hats, right? I just want to say that. But I also want to keep it interactive. Please ask me a question. You're like, I have no idea what that New Zealander is saying. Can you put that into proper English, please? I can, right? So please feel free to interrupt. There's no stupid question. The only stupid thing is when you sit there going, yeah, but what a load of crap, and you walk away and tell someone else that. That's the stupid question. Ask those questions as we go, please, please. This is some stuff we do. We work with pretty much every sector, and what's interesting is no matter where we're at, people say, well, that's all very possible in dairy, or that's all very possible for bison operations, or it's easy for the cropping guys, or it's easy in Western Australia. I had someone say that. Go to Western Australia, see how long you live. Not long, right? Terrifying. And actually, I just had someone say to Megan, well, of course you put on 2 pounds a day. It's easier in Montana where we're at. So we live in a rainfall area of about 10 to 14 inches. We had 27 inches of snowfall late March. It is an extreme environment to live in. We're on the way to Yellowstone National Park. And yes, it's nothing like the TV show, please. Okay. So often I find you think that this doesn't apply to your context. I want you to ask the question of how would this apply? How do we solve these issues in the UK? I'm not from here. You guys know a lot more about your context. You're much better farmers than I am. You understand your farm the best, right? So look at through this lens. So we're going to dive deep into microbials. But what I thought I would introduce too is we have an agroecological coaching school called Create. It's the only one of its kind in the world, I believe. We're running the next one in September at Althorpe. It's a 38-week-long course. If you're interested in coaching, teaching, facilitation in the regen ag space, this is the program for you. We are hoping to get some significant sponsorship for this program. It is not cheap. You get your own coach for 38 weeks. It's an extraordinary experience in delving into this world. So check out the Create course. It's on the Integrity Soils website. I'm going to talk a little bit about myself because it's all about me. This is a photograph of my wedding day. That's Mum, Dad, my brother Jeremy, and my younger brother Mike. Genetically, you can tell that those are actually my parents, right? I have my mother's smile. I have my father's hair. Thanks, Dad. I'm quite grateful for that one. But notice my younger brother looks slightly different. See that? Yeah, so my youngest brother has Down syndrome. My generation is going to be the first generation to not outlive their parents. I want you to get that. This generation that we're in right now are going to be the first one to not outlive our parents. My oldest brother has ankylosing spondylitis. Oof. It's basically like your spine starts to calcify around your main nervous system. I can't talk about it. I won't talk about it. I have non-Hodgkin's lymphoma. I think I'm literally allergic to myself. Can't eat wheat, dairy, wine, can't drink wine. Everything pretty much my producers produce I can't eat, unfortunately. Oat milk, for goodness sake, I have to drink oat milk. Do you know how low that is? All right, but I can't eat meat, so luckily we can eat meat. My brother's also had non-Hodgkin's lymphoma. Both of my parents are incredibly healthy. My parents, my grandparents, there's no history of cancers, no history of any kind of disorders. So you're like, why are we talking about this? because we need to be talking about this, but also because of what I'm going to get into in microbial communications. So my grandfather, my father's father, was 18 when the bomb was dropped on Hiroshima and Nagasaki. And he wasn't able to serve, so he went there to help with the cleanup. Right, so working in that zone and seeing the shadows on the walls and the horror that was World War II. My other grandfather was also in the military. He flew, this is cool, he flew DC-3s and top-dressed superphosphate. Now, in the early days of superphosphate application, what else did they put in with the super? Does anyone know? DDT. Brilliant. And so my grandfather's an incredibly thoughtful individual, very smart, and he feels to this day that he was part of one of New Zealand's worst history in terms of lacing that countryside with DDT. So there's me, very cute. Apparently I was quite obnoxious. And I grew up not on a farm. I grew up as a military brat. So I've never lived anywhere longer than 2 years until I was 28. I've traveled the world. I've lived in different places. So this is the little clicker we'll show you. This is where my house, no, that's not the house. That's the avgas refueling station for the aircraft, right? So this is Whenua Air Base in New Zealand. We could smell it every day. I smell avgas to this day and I feel nostalgic. Oh, it's so lovely, right? This is where the family home was. So my mother conceived my youngest brother when we were living at this place. 4 children with Down syndrome were born on this same road that year to women in their early 20s, and the doctor said that was normal. Just normal, right? We played in the ditches every single day. It was my favorite thing, make little walnut boats and float them down the stream. And what we found now is that water is contaminated with PFOS and PFAS, the forever chemicals. This is the most contaminated site in New Zealand, right? It's actually fenced off from human interference now. You can't even go and look at these areas. And what we're seeing is these substances are showing up around the world. We've been swimming in a chemical soup. It's not funny out there. No laughter. All right, so what I want to talk about is the difference between genetics and epigenetics. So we're all very familiar, DNA, your genetics, that is a duckling. You're not a chicken, no matter how hard you try. You don't get to be a chicken. Right, you're going to look like your mum and dad. That is your genetics. So in the early 60s, when they were doing research on the DNA, what they did was they looked at your X or Y chromosome, broke it apart, took off the protein, threw the protein to the side. First mistake, put the protein to the side, try and understand how did humans do what humans do. But actually the interesting piece of this is the protein sheath that reveals itself to copy the DNA. So your DNA literally is a blueprint. That blueprint says, I look like mum and dad and I am a human being, that you have the same DNA in your hair or in your nails or in your skin cells. So when you murder your mother-in-law, we can track you down. Okay, all right? And so that DNA is the same in all of these cells. Can we open the sides? Is that possible, please? Do we have little fairies that open things? All right. But however, you can have the same DNA, the same code, and someone was drunk the night that they built this particular vehicle. And they went, well, we had the blueprint, but something went awry. What is it that went awry when that DNA is being expressed? So this idea of nature or nurture, more and more research is saying that actually it is the nurturing. It is the environment that we can take these same cells from fat to bone to muscle, put them into different petri dishes, and they will express themselves differently. It's going to turn into bone. It's going to turn into fat cells based on the environment. The environment is the most important precursor for what gene is going to turn on or turn off. So you might have seen this study. I just think humans are just so inventive. We're going to take these poor little mice, expose them to a very strong smell of cherry blossom, and then give them a non-fatal electrocution. Very nice. So every time they smell the smell, we give them a zap. And then half of the mice population were not zapped when they smelled cherry blossom. And what they found was for 2 generations after that, anytime those males, they were IVF, they gave that, impregnated a female, Those offspring, when they were born, if they smelt that smell of cherry blossom, they would shake with fear. That this generation of just emotions is being passed. Trauma is being passed through the memory of our DNA. This trauma is inherited, and what's very interesting is they can actually change that response through nutrition. Are we getting nutrition in our food, people? No. Okay. So epigenetics in people. In the womb, stress, nutrition, and toxins are all going to affect how those genes express themselves. Am I resilient or not? Studies looking at starvation of human populations, obviously you can't do this kind of research, it's not very legal. It's to look at what's happened during World War I and during World War II when populations were starved and abused. What happened in following generations is they were more likely to be obese, more likely to suffer from mental health issues, and their lifespans were shorter each generation. Chronic diseases is the deregulation of your gene expression. So rheumatoid arthritis is a deregulation of gene expression. If you experience early childhood abuse, there's a more likelihood chance of alcoholism, addiction, diseases, gambling. Trauma is passed on, not because you're familiar with alcohol, but because your ability to deal with the world and stresses shrinks. Your epigenome, your gene expression cannot respond to those stresses. You have a very narrow resilience, and I know you know people on this. We can alter our epigenetics, and we can alter that with love. Loving physical touch early on in a child's life can actually alter how those genes express later on. Okay, so we see an increased resilience to stress and anxiety, and increased health and well-being. Nutrition and gene expression, you are what you absorb, not what you eat. Can we get nutrition back into the food chain? Can we find ways to actually switch genes on and off? Working through this and looking at epigenetics in my family, Down syndrome is affected by a difference in gene expression triggered by exposure to chemicals. We're altering the epigenome. Don't think just because I have this gene that means I'm going to have cancer or I'm more likely to have this or that. You can do that on a genetic test. That doesn't mean it's going to express itself. I have the MTHFR gene, which the doctors call the motherfucker gene. Wasn't recorded, was it? Fine. And what that gene means is because I have a double expression doesn't mean that just because I have the double gene doesn't mean that it expresses, but it expresses because of my early exposure to chemicals. So when I was 15, I was exposed to paraquat. I ended up in hospital for 2 days. My neck ended up fusing. I was told I had C1, C2 fusion in my spine. I had headaches probably every second day, migraines a couple of times a month. Paraquat crosses the blood-brain barrier, enters into your spine. It will not leave. It's one of the leading causes for suicide in third world countries because there is no antidote. If you have applied paraquat in your life, if you've sprayed it, if you've smelt it, it's in your body. Right? So I spent another 15 years going to see everybody to try and work it out. Why did I feel so awful? Why was it so hard to get up in the morning? Why did I have foggy brain? And finally, met somebody who diagnosed it, and we did hyperbaric chamber, intravenous vitamin C, homeopathic support to support me through while this gray stuff that stank like paraquat came out my nose for 10 days. It was disgusting, right? You guys are laden. That means now, because my son was born obviously after, I didn't have a son at 14, smart about that. All right, so my son has different epigenetic expressions. He has ADHD, he's just the kind, right? He's here to just test me, he's brilliant. Through ensuring he had nutrition and he wasn't eating things that he was allergic to, we've managed to not have his genes deprogram, right? He's 6'3", he's incredibly healthy, and we did that through making sure that he was getting the nutrition that he wanted. Hey, blokes, can you move away? We love you. Somewhere else. I have no filter, you might notice. That's also a genetics. Sorry, okay. We can heal this This is not a death sentence. My brother manages his ankylosing spondylitis through diet. He finds if he eats maltodextrin, it fires it up. He was noticing every Friday he wouldn't be able to walk and he couldn't figure it out. Every Thursday night he was eating barbecue chicken, sneaking out because his wife is vegan, so we have to sneak out, go and get our barbecue chicken. And then he started to realize, I have this pain on Fridays. Why is it just on Fridays? Tracked it down to maltodextrin. It's in all of these processed foods. We've got to stop eating processed food, heat-treated food. You guys know all of this. That's why we're here. How do we get nutrient-dense food in? How do we get whole foods in? So if you want to reprogram your DNA, take a photo of this. This is the Whole30 program. Very simple stuff. Reduce and eliminate your allergens. Please take an IgG/IgA blood test. I wish I had done this so much earlier. It picks up your chronic and your acute allergies. I sent it away to prove to my husband and son that because they were so obnoxious, it was because of their allergies. They came back in the green and I was everything in the red. It was very disappointing. It's the opposite of what I thought was going to happen. Drinking clean water. That tap water you got's not clean. How do we get clean filtered water without just buying bottled water? Infrared therapy. This is the interesting piece, is looking at what were traditional people doing. What were the Celts doing? What were ancient indigenous civilizations doing to actually repair their DNA? So hot and cold therapy, sweat lodges. Talking to my Lakota friends about how memories are coming back. They haven't lost the memory and the wisdom of working with land. It can be released. Those memories can be released through a process of like sweat lodges, of initiations for young people. Where are those initiation moments that are actually reprogramming our DNA so we're not passing that trauma forward? I use fulvic acid all the time. Fulvic acid when I'm traveling. You have chemicals in the water, you have chemicals on your food. Even organic food, unfortunately, has some chemicals on it unless it's being managed regeneratively. Not to beat up on organics, but we need biology on those leaf surfaces. We need biology in the soil to break down chemicals that are coming down in the rain or in your irrigation, if you're irrigating. So we need to take a fulvic acid or something similar to bind to and then eliminate. So 90%, I think it's over 90% of Americans now have glyphosate in their urine. I did the test, I'm 0. I don't have glyphosate in my body because I take a binder, all right? Not a nutritionist, you can ignore me, whatever, that's fine. What are you talking about? Keep going, woman. What's it got to do with soil? All right, so how does this affect gene expression in plants? The different types of soils that you have are altering the expression of genes in those plants. Climate alters gene expression. Am I going to switch on a gene that's going to enable me to deal with hot, dry, wet, cold? The amount of organic matter will influence how a gene is turned on or turned off. So we inherit those traits. So what we're seeing and talking to plant breeders is how do we prepare plants for an uncertain world? How are plants going to be able to deal with these extremes instead of what we currently have, which is how can I grow a plant with as much NPK as possible and all the fungicides and pesticides? We don't want to be dealing with seed like that. Our biological relationships. There are plants now like wheat that will no longer communicate with mycorrhizal fungi. Why is that a problem? Sorry? No food. Well, the plant will grow, but you're not going to have adequate phosphorus, you're not going to have zinc, you're not going to have the secondary metabolites that I'm going to talk about if we've interrupted this relationship, right? Heavy metal stress, that's all about epigenetics. So microbes, mycorrhizae, your fungi, your bacteria can actually alter the epigenetic response in a plant. They can turn a gene on or turn a gene off if you have the microbes in the system. So the microbiology in the plants are communicating all the time. How do I deal with an uncertain world? So how a plant is growing and where you're sourcing your seeds, this is critical. Is that growing in an environment just full of chemicals with a very minute seed microbiome? We're learning more and more about the value of microbiology inside the seed. So it can either set you up for success or failure, especially as we're starting to move into low input systems. We have plants and seeds that are not adapted. On the, if I say left, that's the little one for you. It's the same thing. Magic. All right. On the left is, I don't have a word for conventional. industrial managed? What do you guys call it? Chemical. All right, so chemically managed fields on the left, that's wheat. On the right is kamut, same age, planted on the same day. Kamut or Corazon wheat, older varieties. Taking a look, what's happening with those root systems? Have we got that root mass? Have you got those rhizome sheaths? This is the difference between drought or not. We're not breeding plants for massive root mass. We were focused so much up on top in terms of production. It's kind of like breeding horses, race horses, to race fast, but you forgot that you needed good feet, and they're all pricks. All right? So, personality you might be breeding for, when what you want to look for is, have I got resilience? So we're not going to breed for a single trait. We're breeding for, have I got a resilient system? Can this plant communicate to the microbiology that it needs? So stress is going to alter your gene expression. Pesticides, herbicides, soluble fertilizers, neonicotinoids alter 600 genes in the plant. 600 genes are altered just through the exposure of these chemicals, and those genes are required for disease and pest resistance. It's a great sales pitch. Buy my product, you're going to need this other product. If we have poor nutrition, so low functional calcium, and I'm seeing low calcium even on chalk soils, what is happening with calcium? That's all about beneficial fungi. Nitrogen, boron, copper, manganese, zinc, what is happening with those elements? They affect that plant's ability to express genes, and they affect human health in the same way. I can't turn on these aspects to build my immunity. If you're cultivating, overgrazing, waterlogging, doing all the things that, like waterlogging, right? You think that's a natural event. We've had some massive rainfall events. What I want to see is, yes, you might flood, but how quickly do those fields dry out? Do you see a field that acts like a sponge? Are we holding onto water, or is it stinking and sitting underneath that? Right? That is all about gene expression. So I want you to think about anytime there's a stress applied to the plant, different types of genes will respond. And that's going to depend on what's the phenotype, what's the breeding, what's the nutrition, and how can that plant respond to stress. So then what we find is that alters your root exudates, the expression, the composition of our primary and our secondary metabolites. And we're going to talk about those. So your root and your rhizosphere, that area around the root, is where you get all that biological diversity and the biomass. These are the functional groups. The more that that plant communicates, and it has the ability to communicate, the more diversity we see in the soil system, the more the plants communicate with the microbes, and the microbes communicate back to the plant. Right? We see a plant's ability to respond to stress. I just want to give you one example of what this looks like. So ergothioneine. Guys are all familiar with ergothioneine? Does anyone take it? Want to maybe get interested in this one. All right, so it's an amino acid produced by fungi. You will find it in, interestingly, not in button mushrooms and portobello, but like your oyster mushrooms, your reishi, your lion's mane, are full of ergothioneine. This amino acid's been called the longevity vitamin. Why are they calling it that? Because it's linked to Parkinson's disease and Alzheimer's and reduced life expectancy if you don't get adequate ergothioneine. Yes? Oh, that's a good question. I don't know. I think you might. But it's for yeast byproducts. Good question. Google it. Okay. Not advertising. Okay. And so what's so interesting is this is a secondary metabolite produced by fungi. They pass that on to the plant, and we can measure it in plants. If you're tilling your fields, the ergothioneine disappears. And this study was just done in tilled fields to conventional fields, conventional no-till chemical-used fields. They found there was a significant difference. We haven't done this research in regenerative. What does it look like if everything that you do is fostering fungi? Can you till? Yes, you can. How do you earn the right? We're going to feed fungi. Every time we're out there, we're thinking, how do I feed my fungal biomass? How do I encourage more and more diversity of biology in the system? Hello? Oh, wrong number. Very good. Okay. So, you've talked about this a lot. What are the soil health principles? You guys have had this jammed down your throat a few times. What are they? Keep it covered. Living roots at all times. Diversity. Integrating livestock, and that could be biological livestock. That doesn't have to mean you have to run cows in your garden. Okay, reduce disturbance. I talk about optimizing disturbance. Disturbance is actually a natural phenomenon. If I'm disturbing, how do I optimize that for living biology, right? You've got the principles. So every time you're out and about, we also talk about a collaborative mindset as being a principle. Am I looking at my landscape and thinking, how do I get in sync with nature? How do I increase more vibrant life in this system, right? And using that as a principle. So if you follow principles, slightly higher chance you're going to be regenerating. Doesn't necessarily mean you can, right? I've seen people who are keeping ground covered, putting in cover crops. They put in an additional, correct me if I'm wrong, but maybe like 50 million acres of cover crop last year in the USA. How are they terminating that? Yeah, yes, great plan. Terminate it with glyphosate. We're going to increase our glyphosate use last year, which is what happened, right? So think about, how do I do this in a way that I'm fostering microbiology? Maximizing photosynthesis. Your main role is to plug into the sun. How well is my plant capturing sunlight energy? Do I have a diversity of solar panels in there? Am I capturing every piece of sunlight energy that I can? And what I'm seeing in the UK situation is mostly, and I haven't been to a lot of places, but your photosynthetic rate of capture is very low. And I suspect it's due to minerals. It's due to trace elements. Take a look, get your diagnostic hat on. Why are my plants not capturing sunlight energy? Because that driving of sunlight energy, pushing sugars out the root zone, is what's driving all of these benefits. And we talk about secondary metabolites, you'll see why. Okay, very good. Context, just briefly, very important. I don't want you to cookie cut and try and do exactly what someone is doing in New Zealand or someone in Montana or someone in Western Australia. What is your particular context? What is your challenge? And again, what does it look like to have a fully biodiverse environment for yourself? Living roots, diversity plants, that's fine. So increasing plant photosynthesis, you need to be addressing plant nutrition. What is happening? Start doing leaf tests or sap analysis and take a look. Does my plant have what it needs in order to do its job? Because when plants are low in nutrition, what they do is they actually shut off capturing sunlight energy and photosynthesis due to sunburn, right? They're going to get burned from all those photons. They'll actually stop photosynthesizing. You'd be like, but the plant looks green, it's fine. It's just getting through life. It's on the welfare system. But it's fine, is start taking a look. Right? Is there something that's undermining your plant's ability to be fully expressed and capture sunlight energy? Reducing plant stress. If you're not sure if it's your management, it probably is. Right? Why are my plants stressed? Have I been overgrazing? What's the definition of overgrazing?
Time.
Sorry? Time? Maybe? Time? The second bite. That plant starts to recover and someone comes along and bite it. New Zealanders are the best rotational overgrazers in the world. We have it down to a fine art. 14 to 21 day rotations. We're going to keep it humming. Those plants, when we test them, super low bricks. What you're feeding are non-structural carbohydrates. These are the stuff that cause inflammation in animals and horses. You'll get navicular, you'll get hoof issues, inflammation, somatic cell counts. Right? Those plants are stressed, they start to shed roots, they are no longer to invest in root systems, and they're certainly not feeding microbiology because they're just getting through their own day. And then what you're going to find is you're going to lack persistence, right? You're going to have to come back in and oversow and overseed and all that. We want to build functional soil carbon. Some of you have very good carbon levels. It's not functional. These are the soil systems we call constipated. If you go to New Zealand, rampant diarrhea. Okay, you choose. Can we get that functional functional soil carbon up? It's all about optimizing biology and these beneficial metabolites. Have you seen that thing with the underpants, soil your undies? Right, what's breaking those underpants down? Biology eating it, right? It's actually not a measurement of biology, it's a measurement of enzymes. It's a measurement of the secondary metabolites being produced by biology. It's not actually biology doing it. The workhorses in this are the secondary metabolites in our enzymes. So these are the plant primary metabolites. So this is a root tip. That plant is exuding that material into the soil system. The primary metabolites are fats and proteins, sugars, right? The first elements that are made by the plant. They're stimulating and feeding microbiology and sending signals, right? You're going to stimulate a whole lot of microbes when there's a whole lot of food in the system. Is it just me? Is it hot in here? I'm dying. All right. So yeah, we can look at these processes. So think of the short carbon cycle is all of the material that we're adding into the soil. That might be these fats and proteins. It might be compost. It might be compost extracts, poos and wees, animals trampling, stubble. This is the short carbon cycle. Very important. The soil breathes in and breathes out and breathes in and breathes out every day. What we're interested in is how do we drive the deep carbon cycle? And I want you to be measuring, those of you doing carbon, I want you to measure organic carbon and inorganic carbon, because fungi are literally making rocks. Permanent carbon sequestration if we can harness the role of fungi in the system. Mycorrhizae, myco meaning fungus, rhizo meaning root. They live inside or on the plant roots, and they are able to get far further out into the soil environment than roots can by themselves. Plants without mycorrhizae access about 2% of the soil volume. With mycorrhizae, it's about 20% of that soil volume that they can now reach for water and nutrients. Don't tell me you have a nitrogen problem or a phosphorus problem. You have a biological problem. Don't tell me that there's no zinc in your food anymore. It's a mycorrhizal problem, right? Don't tell me you're in a drought. It's a microbial problem. Go and test. Have I got mycorrhizae in the system? We're testing properties and finding less than 1% mycorrhizal colonization. You have a problem. We're trying to figure it out in rangeland, actually, so extensive rangeland. Why is it we have no mycorrhizae? Plants need it to survive. The researchers I'm working with suspect it's the anthelmintics. It's the drenches are disrupting the role of mycorrhizae, because there's nothing else out there on these rangeland environments. If anyone else has an idea, I'd love to hear it, but if we're overgrazing, stressing plants, they're not going to invest in mycorrhizae, they're not going to invest in their microbial community. So consider the plant outsources its functions. You have everything inside your body, and you can go and walk down and get an amazing looking pizza that I cannot eat. It looks really good, is that you can walk yourself down there, take your high digestive tract with you, your brain and your ears and your eyes, and you can source materials. Plants can't do that. Think their endocrine system, their gut system, all of it is outside of its body. It's outsourcing these functions. So imagine underneath the ground, there's a total underground metropolis. There's a city. And through that city weave your earthworms. What's building the smallest bricks is the bacteria. What puts the bricks together is your fungi. We have protozoa using the bathroom. We have nematodes coming weaving through up and down corridors. We have structures. We have hospitals, schools, and a pub. We do. And what we find is as we start to degrade that soil structure, you lose your hospital, you lose your school. What happens to the community when you lose your pub? Revolution, riots in the streets, exactly. So what we're seeing is we're losing these services, these soils start to collapse. 80% of New Zealand dairy farms across New Zealand were surveyed, significant compaction. We've lost these services. Now you're going to need more nitrogen, and more nitrogen's being put on. So our secondary metabolites, consider these are natural products predominantly made by bacteria, fungi, and plants. And they're releasing them all the time. Close your eyes right now. Just close your eyes and imagine you're in this underground metropolis. Eyes are closed. How do you know how to get around? How are you going to get down to that great pub? How do you know who wants to eat you? How do you know who's your friend, who's your food, and who wants to make love? Open your eyes. How do you know? You don't have eyes, no ears. Smell. You don't have a nose. Feel it. Sense it. Our senses, right? So these are the secondary metabolites, is what the plants are using to communicate with each other. So they're not directly involved with the normal growth and development of plants. So it's why there hasn't been such a strong focus. NPK grows big plants. No one was focusing on what does this mean for human and animal health if they're eating that food. Do we have ergothioneine in there? So Your secondary metabolites include things like your phenolics, your alkaloids, essential oils, flavonoids, the stuff that gives our flavor to our food, the stuff that gives you your terroir of your grapes. It's not glyphosate, people. The terroir of glyphosate. All right. Drugs, antibiotics. So pretty much you get high on, you smoke, you just like that was quite pleasurable. Probably a plant secondary metabolite that you're experiencing. Terpenoids, coumarin, peptides, lectins, glucosinates. So what plants are using to communicate with and what we need in our own gut system. You don't eat your food. Do we have Anne in the room? Beautiful Anne, you are what your food ate. Go and read her book. It's these secondary metabolites, what you eat and mostly what you poop is bacterial necromass, right? You're absorbing their secondary metabolites and their byproducts if they have nutrition and if they're healthy and if you're not full of something like yeast or candida, which I think many humans are, or you don't have leaky gut, all of that, right? So these are the things that are giving that plant health and nutrition. It's what gives you flavor and smell. They did a study recently in the US and found that flowers are no longer sending out scent due to the amount of smoke and smog. They're not investing in smells anymore. The land gardeners are crying. No beautiful smelling flowers, because those are secondary metabolites. They're not going to invest in it if no one can hear the signal and no one's listening, because they drop out with the exposure to smoke. We need nutrition in order to form these secondary metabolites. What's happening with nutrition? So consider that the cell membrane, every single cell wall is 100,000 receptors waiting for a signal. So there's a physical awareness of the environment that you're in, and then there's these signaling molecules, so that each cell has a receptor cued into different types of signals. So those signals are received by those receptors. They pass through that cell membrane, either through the channel or the signal transduction. I'm in a hot, cold, sexy environment. Right? Or just a hot, sexy environment right now, right? So that changes how that membrane receives those signals. It changes what gene is activated for behavior. It changes what gene is activated for me to release this type of signal, all right? Or this kind of response, and I can now respond to the plant, right? It activates the cells. Your primary brain is every single cell wall. It is not this bit of gray matter, and it's not your gut. It's every single cell is waiting for a signal. What's the environment that you're in every day? Well, I'm hyper-stressed. I'm just super stressed out. I'm overwhelmed. You're changing your environment to every single cell in your body. Those plants are super stressed. It's changing their environment and how they can signal to microbiology. So one of these mechanisms called quorum sensing, you might be familiar with that. This regulates the gene expression of certain types of organisms. It's basically what plants are using and microbes are using to communicate and to coordinate group behaviors. So it was originally discovered in the 1960s, looking at why is it that you'd get the bioluminescence at the beach. What enabled those microbes to signal, the phytoplankton to signal to each other? It's a chemical signal. Quorum quenching is when that signal will be turned off. So at any one time, you could have strep organisms in your body, but you're not feeling an infection. You've reached a certain threshold, and now you have virulence and you're sick. But it's reaching that threshold of a biological population. They switch on a gene, and now they're virulent, and now you feel awful. So what they discovered is there is a chemical signal to flush cholera out of the body. Parts per trillion you can drink this liquid with a single drop. So parts per trillion is 1 drop of water in a swimming pool. That's how dilute these chemical signals are. And if you drink this drink, the cholera will be flushed from your body. What's fascinating is that chemical signal is the same one to turn off botrytis in grapevines. Parts per trillion. Wild. I found some interesting research and talking to my Lakota friends about watching buffalo and how far away things like dung beetles would know that the bison were coming, right? And it's about 2.5 miles away. A dung beetle can see a dung pile that might be bison or manure from 2.5 miles away, and it looks like a fire in the distance. They can sense that from the secondary metabolites. And they swarm in if you have a healthy ecosystem. right? Parasitic insects, they're listening for the signal. And what the signal is on a lot of properties is literally go away. Don't want you, don't come. And so we're seeing these drops, dramatic drops in insect populations. This is what it looks like when dung beetles smell nutritious dung. This property, when we started working, had not seen dung beetles for 65 years. I'm going to play it again because it's my favorite video of all time. 700 dung beetles in a single cow pie. We counted them ourselves. We also got another cow pie, sent that away to entomologists, 700 dung beetles in a single cow pie. They talk about following the bison, and the manure was already gone hours later. Right? That microbial, biological system works so quickly if they're getting the signals for health and nutrition. All that happened on this property is we started to do some of our soil amendments, and we started to feed cattle raw humate. Right? That raw humate is full of 65 million year old secondary metabolites, proteins, and signals. It's wild, wild. All right, so if that plant comes under some kind of pressure, it's going to signal to different types of microbes. They recognize that, and then they produce things like our quorum sensing molecules, your antimicrobials, what enables a plant to defend itself. They produce phytohormones, how a plant is able to grow. And they produce volatile organic compounds. These are the things that can move through soil and move through the water and communicate from plant to plant, from plant to microbe, microbe to plant. In response, the plant now can synthesize plant hormones. It can respond to stress. It can synthesize flavonoids, defend itself, immune suppression, processing proteins, and that was the primary metabolic function. The piece of this is the plant doesn't do it by itself. Its gut is outside of its body. Its digestion systems, its communication happens outside of those roots, and in order for us to produce top quality food. It's a microbial bridge, and they're secondary metabolites. So I've just used one example, salicylic acid. You're probably very familiar with this one, the plant protector. This is probably the oldest known secondary metabolite to be used. It occurs bacteria, fungi, and plants. Willow, so doing your willow root extracts. Wintergreen, birch trees, mangoes, meadowsweet, guelder rose, all very, very high in salicylic acid. It's very important for plant growth. It's what helps to defend against pathogens and buffer against environmental stresses. Parts per trillion. So I know people want to put aspirin in their water when they're watering their seedlings. That's far too concentrated. Parts per trillion is what the plant requires. So if you imagine plants coming along and hit compaction, people think compaction stops roots. It's not. Those roots can push up tarmac, right, and push up a road. That physical penetration of a root through compaction is not impeded because it's tight. It's impeded because of the production of ethylene. So what happens is we get ethylene buildup underneath those layers. It can't off-gas, so ethylene is produced by microbiology. They can't off-gas it. It builds up. Now whenever a plant is extending down and trying to move through that system, it sheds the root hairs and the root tip. So if you ever put apples in with your potatoes, apples, yeah. So an apple releasing ethylene can actually stop that production of root hairs. So we use it for ripening, but it will stop the production of root hairs. It stops your potatoes from sprouting. You're like, I don't remember what that New Zealander talked about, but I went and put an apple in with my potatoes. It's fine. Okay. All right, so what we're seeing is these biological systems going on. Salicylic acid, when applied to the plant, helps to protect the plant against ethylene, and we get root penetration. So doing things like root transplants or seed transplants, putting salicylic acid on it as some of our soil treatments. And I'm only using this as one example of tens of thousands, right? But there's more research has gone into salicylic acid because of its effect. So it alters microbial diversity when you apply it. It'll actually change from more, not necessarily just benefit, like pathogens to beneficials, but there's a sway in the microbial community. It suppresses disease. It's really good research in Phytophthora, Fusarium, Botrytis, viruses, and powdery mildew. Just small amounts of salicylic acid signaling to the plant, priming those plant defense systems, it also suppresses pests, particularly sapsuckers. You guys have got sapsuckers in your pasture. We've got them in crops. If you have a low Brix, so low photosynthesis, below 12, but generally below 6, we're going to see the sapsuckers coming in, right? So this might be something you can add into your program to help to defend your plants. And this could be simply by making compost with willow, right? Or mixing willow wood chip in with your compost or your extracts or whatever you're making. Is just get a little bit of it in there. So let's imagine this plant is growing without the presence of trichoderma. Who's using trichoderma? Ah, good. All right. In the last talk, there was actually Andrew was talking this morning, so he's using trichoderma. Trichoderma is a fungus that more specifically attacks disease-causing fungi, so using it for the control of Phytophthora, Sclerotinia, different types of fungal diseases, and in this case, botrytis. So if you imagine you have something like botrytis showing up, and that plant is signaling to its microbial community, specifically to trichoderma, hey, dude, you're out there, I need some help, I need the fire engine to come in and put out this fire. No one's responding. When you have the presence of trichoderma, what we see is the plant signals to trichoderma, and the trichoderma responds and primes salicylic acid, abscisic acid, and jasmonic acid in the plant. The plant can now defend itself. It can't do it without the presence of certain beneficial microorganisms in the soil. You with me? You're like, can you repeat everything? It's fine. Okay. So salicylates for human protection. It reduces the risk of multiple chronic diseases in humans, including heart attack, stroke, arthritis, diabetes, certain types of cancers, and Alzheimer's. So this is new research. New reviews are looking at the role of salicylates, which is one of the byproducts of salicylic acid, in foods. If we are undermining the microbial communication and the biodiversity underneath our feet, we're undermining our ability to defend ourselves. We're undermining our gene expression in the human population by degrading our microbial communities. Do you get the impact of that? Like, really? Where are the secondary metabolites? Oh, you got lots of protein. Awesome. So we're dying of these hidden hungers, right? Lots of food, and we're starving. Right, you've got all the food that we possibly could, but we're missing these secondary metabolites, we're missing our trace elements. There is a problem. So where do we get these metabolites and biostimulants? There are rich sources of it. This is my compost, my vermicast. So this is literally vermicast, right? So I put worms through that. You can see the wood chips. I want to see beautiful wood chips. I want to see fungi growing in that medium. They're releasing secondary metabolites so that if you make an extract and you beat this up and you kill all the hyphae, it doesn't matter. Those secondary metabolites have a much longer shelf life. So I'm less concerned about the living biology, and actually I think it's dangerous. So if you're putting on single inoculants, so a good example of this is in the 1960s, they developed a rhizobia for soybeans in the US, and they planted all of those soybeans with that inoculant. Now they've developed an even better rhizobia that makes more growth and superpowers those soybeans, but every time they plant the soybean with this inoculant, the original rhizobia outcompetes and suppresses yield. If you are buying mycorrhizae, they're probably made in India, just saying. Right? You're bringing in non-native organisms into these environments. So I'm more interested in how do I stimulate my native microbiology, and how do I provide the metabolites and the nutrition so the plant can dictate who's thriving, who's surviving, and what's happening nutritionally, right? So we want diverse microbial-rich products like these, fungal. So this could be a Johnson Sioux. I see there's people making Johnson Sioux compost. Even on a large scale, if you are in arable, one of those single Johnson Sioux is often enough for your whole property, right? You don't need to make a lot. We're making a very, very top quality product. Natural Korean farming is a very rich source of these metabolites. Mushroom compost, watch it, because sometimes mushroom compost is made and then they sterilize it with methyl bromide. Delicious. All right, so don't buy that. So make sure that if you're using mushroom compost, it hasn't been sterilized. There are natural hormones, so willow extracts, seaweeds, There's a bunch of other things, right, that have phytohormones. So some plant extracts will be high in our hormones. We have enzymes, barley ferments. So if you're picking up some of these byproducts from, say, beer production, there are commercial products on the market that are super expensive, and all they've done is ferment barley, right? And the minute that that barley seed, the harder the seed, the more enzymes it needs in order to crack that seed hardness. So that minute that that starts to germinate, you've got this massive concentration in enzymes, and people are selling it for crazy money. You can make it yourself just by fermenting barley, right? Broccoli sprouts also. So feed this to your chickens or your self. Do that. All right. And then different types of amino acids and proteins, all right, are hugely stimulatory. We do use quite a bit of fish hydrolysate. I know it's probably not sustainable, but they're not killing the fish for you, right? It's for human, it's a byproduct, okay? And then some of your plant ferments. Okay, so lots and lots of ideas about plant ferments that you could make. Biodynamics, some of these herbal extracts are full of enzymes and secondary metabolites that are going to help to benefit what you're producing. And like I said, I'm a big fan of humates. Hello. Goodbye. Right. The more diversity you have in a system, the more diversity of plants, root architecture, and families, they are signaling to different types of microbial groups. They're waking your soil up. They're putting those metabolites out, and they're lifting the health and the health of what you're actually producing. So always think, how can I add more diversity into the system? And secondly, the diversity of what you're doing, or what you're doing above ground, is going to alter that native diversity that's going to germinate. And we're going to talk about that in the second session when we talk about plants as indicators, is how can you tell by what's germinating, what's happening with some of these signals, if you guys are still here, because it's a long 3 hours with me. Good luck. You're going to hear my accent in your sleep. Okay. So one thing you can do is do a foliar test of some of these products that are coming out. Right? So what you're going to do is you're going to set up just 1 meter by 1 meter. And I know you weirdos do yards these days, like yards and miles per hectare. I don't know what you're doing. Okay, so 1 yard or 1 meter squared. What I want you to do is try some of these products in a spray bottle, spray that product at its recommended rate. So if you're doing 40 liters per hectare, that's 4 mils per square meter, right? Because you're going to divide 40,000 mils by 10,000, right? So easy math, nice easy math. Thank you, John. I don't know what to do without you, right? Okay, so by doing that, you then wait for 40 minutes and you're going to come back and test it. All right, did this seaweed actually lift my plant photosynthesis? Did this product that I'm buying that's all these different things, is my plant extract, is my compost extract lifting the photosynthesis of my plant? If not, you want to maybe leave it for a few weeks and come back and just see, did I put too much on? So I was working with a dairy farmer who had the most beautiful, lush, huge grass sward, and his cows looked like the Humane Society was going to come and put them down, right? Just skin and bones, milk production crashed. He's like, I don't understand. We're using this product and it has seaweed and humate and fulvics and yum, 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 right? And you're going to see these products. And very small fine print at the bottom, it said gibberellins and auxins. What are those? Plant growth hormones. This company was packaging plant growth hormones with biologicals to make it look fancy. The bricks, when we applied it, dropped by 3. Plant photosynthesis and these little trial plots dropped by 3. He's growing basically empty calories. Those cows were eating, what those hormones do is basically cell wall elongation. It's empty cells. I know the gibberellins make you feel better in the middle of winter, but they're going to rob your calcium from your soil, and you're not necessarily growing anything that's more nutritious. It's just better. I know, you look out the window, there's grass, yay. But, so do this, really, really simple. So start to take a look, because you will see snake oils turning up, right? We do see products that genuinely are not going to be of benefit, right? Take a look, is that an enzyme? You know the deal, right? Just give it a go, see what happens. And then wait a few weeks. So we've seen these effects with good management last for years from single applications. Try the milk. So you can try a little bit of milk. Milk's a huge biological stimulant. You can use milk powder. You can use pasteurized milk. You can't use 2% milk because it's not milk. Just so we're clear on that. Okay. And no oat milk. Dear Lord. I don't know what that does. I don't want to know. I got to have one vice. Cups of tea. One vice. All right. So I want to show an example. How am I going for time, buddy? Where's my time, adult? 15. Perfect. Oh, I'm amazing, just so you know. Okay. So, right. We got these metabolites in action. So this example here is from a vineyard in New Zealand, Kokako. This was actually my first client. Absolutely terrifying experience. So 2006. He's like, you need to come and consult. I'm like, I do not want to tell you what to do. As much fun as that is in my family, I don't want to do that for others, right? So this is what some of his rows look like these days. So there's a nice mixture of what can you see growing? Buckwheat. Yeah, what else? Phacelia. All right, I got something for you. You're never going to be able to say this again without hearing this. Phacelia, I'm down on my knees. I'm begging you. Sorry. Okay, so every time someone says Phacelia, I hear that stupid song. Anyway, that's from John O'Frew in New Zealand. It's his fault. So diversity. That's one of the things. So when we started working together, traditional viticulture, the land had actually been in spuds, particularly, mainly potatoes. And what they found was they were having to apply 6 herbicides a year, and they could not control their weed problems. They were basically growing cask wine, very high fertilizer inputs, very high disease pressures, stressed problem blocks. We did not count a single worm, stressed viticulturist. They're making absolute bollocks wine, it's a technical term, and not making any money, right? So he's like, look, you can't do anything worse than what's been happening so far. And I'm like, that gives me such confidence. I think I can work with you. So in 2007, the driver was increased costs, reducing returns, very poor visual assessments. They had huge vigor blocks in some areas, low vigor in others, very uneven growth across the blocks, terrible wine. So what we did is we introduced some regenerative practices. We started to apply a metabolite product, no living biology. This was all secondary metabolites in a concentrated brew. We would do 10 liters on the ground, and then as a foliar, 7 liters 4 times during the growing season. We changed the mindset, very important piece. Please change your mindset. Hardest thing to do. It's the hardest piece, right? Start looking at how do I bring more life into this ecosystem. He reduced and buffered any actions which are harming soil microbes, removed all those organophosphates. He was like, I don't want to be responsible for something that's killing my staff. So I think it's a good start. Stop killing people. Marvelous. They also buffered any of their glyphosate. So they were still using some glyphosate. But in 2010, pretty much the herbicide was removed in nearly all the areas except some cultivars, and moving to undervine mowing. Within 1 year, we went from 6 herbicides a year to a single herbicide in blocks where we couldn't control the weed. The metabolites changed the signal underneath the soil, and we no longer saw these very tall, weedy, herbicide-resistant plants showing up. I'm going to talk about it in the next session about what do we do with herbicide resistance. For me, it's very simple. It's the biggest problem facing agriculture right now. Well, apart from the mindset, but that one. Okay. So this is what the vineyard looks like now. So you can see how much grass they've got underneath that. Beautiful. So this metabolites is sleeping compost tea. It's a 6-week brewing process. They start with native soil and compost. This is a commercial product, but you can make it yourself. They're feeding them a range of biological foods, macro and trace elements right through the season, right through the 6 weeks. So you've got a big brewing tank, and you come in and you feed a small amount of this and a small amount of that. And every day you're kind of mixing it up, giving these microbes different foods so they go through bust and boom, but keeping it aerated, right? That sprayer there is from Chaos Springs. It's a slurry sprayer. I highly recommend you start looking at getting some of these types of machinery. This thing can spray up to a 1/2 centimeter size material. I can put my compost straight out. I can put minerals, up to 60% minerals will go into these tanks. I can spray it straight out. I can put seed in it. Versatility, very easy to build. These are small ones. We have ones in Montana that are, so that's 1,000 liters, right? I've got to put it into English. 4,000 liters size sprayers, right, that we're using on rangeland. So the results, what he saw was a massive reduction in inter-row variation. The ripening became very consistent. They no longer had to do anything about their yans. I don't know if you know this, people, but the wine that you drink is no longer fermenting naturally. They're adding urea into the tanks to brew their wine because that microbiology is no longer on the leaf surfaces. There's no nutrition in that stuff anymore. Buy your wine from regenerative operations that are not feeding urea into their wine, all right? They also so it's super vigorous area. We saw reduction in vigor. Areas that were low vigor saw an increase. It's just the amazing evening out effect of what we see with microbiology. They also eliminated, reduced all nutrient-based deficiencies. They had a lot of manganese disease. What's manganese disease? It's glyphosate. So glyphosate binds up your manganese. If you drive around vineyard areas and orchards, you see this yellowing. The yellowing is due to glyphosate. It's binding up their manganese. So these guys no longer needed to apply manganese, no longer had water stress issues. They actually were able to pull out their irrigation systems. What would that be like for you if you didn't have to worry about water? Massive reduction in mealybugs, and we saw earthworms going through the roof. So overall, the consistency in fruit, improved flavor, excellent yans. They went from cask wine to super premium wine. These guys are now making money. And they're happy. Yay, right? We want that? Lovely. So they've expanded to 100 hectares, under vine mowing mostly. They're using half the sulfur, and no botryticides have been used in 14 years. Botrytis is gone. Botrytis no longer shows up in these vineyards. And no penalty price drop on bricks, if anyone understands that. They got surveyed by MPI, so the Ministry for Primary Industries in New Zealand, which is now, I don't know, they change names all the time. Do you have that? Like your Prime Minister, right? So it's a government agency that works in agriculture. They found they were the most cost-effective vineyard in the North Island that they surveyed. So they surveyed right across the country. Can we make money? Yes. Can we do that and grow quality? Yes. Can we do that with much less stress, much less tractor passes? Oh, yes. But we've got to get interested in what's going on. How do I stimulate microbiology? Okay. We're going to break in a second. So any questions about any of that?
You were previously talking about the epigenetic effect of the conditions on seeds and plants. In systems that are retaining their own seeds, if that seed has gone through a drought or another stress factor, do you see a change in the outcomes in the next crop?
Yes, so not only if we can change that, the outcomes can actually be measured in multiple generations. So typically this might be in 2 generations, it could be in 5 generations. You've altered that plant's response to your natural environment. So it's why we want to be keeping our seed. But they did some study with nematodes and found that, oh geez, I think I got the number wrong. 14 generations were altered from that single gene alteration through the females, right? So if you're altering gene expression in your seed, keeping your seed, you are creating locally adapted plants.
Sorry to...
Oh yeah, I'll let you go. Yeah.
So if you have a really good year,
yes.
should you hold back more of that seed to use in other years? Say the next year's a drought, are you better off using the seed from the good year?
So if we have a really good year, are we better to hold back the seed from the good year? No, you're better to hold back the seed from the bad year that thrived. I want plants that were really thriving, the outliers, get interested in that. If you've got lower yield overall in that bad year, maybe not, right? So look for the plants that were really thriving and really had their genes switched on.
If we buy your book,
Yes,
For the Love of Soil,
yes,
will we get all the bits that we didn't take a note fast enough and my brain will have forgotten?
yes. Yeah, so this
Thank
case
you.
study's in the book, but it's also on my website. And Epigenetics is the book I'm writing right now, so... Oh, God, I know. It's a piece of string question, that one. Hopefully soon. We'll see. Life keeps getting in the way. But, yeah, don't buy one book. Buy 5. Buy them for your friends.
Have you tried the treatment on honey fungus, the celiac acid, the willow treatment, the willow drenching that you've done on honey fungus at all?
Like amalaria?
I don't know I just know it's the root of quite a lot of our tree diseases right now.
So amalaria, I've had a great time with it using compost extracts very successfully. So amalaria, take a look. So a lot of these things like a root rot, a phytophthora, amalaria are secondary. They're not your primary problem. What is underneath that, and it's generally nutrition and not having adequate predators and adequate microbiology to protect it. So don't blame the dieback. Get interested in why is that plant not able to thrive.
We grow 3-year herbal lays in an arable rotation for forage production. When we come to destroy those for the next crop, are we best to use glyphosate to start to kill it off, or are we doing more damage by ploughing? Which is the best thing to do?
It's the $1 million question. You could do either. You need to buffer and then you need to repair what you're doing. So earn the right. So if you're cultivating, I want you to put biological foods down while you're cultivating. Some humic acid, some slow release carbon forms. If you are using glyphosate, I want you to put fulvic acid or worm extracts with it and drop your rate. And you didn't hear that from me, Bayer, right? So you can drop it by 30%. So I'd say most of my cropping, so we work with about 300,000 acres of cropping, and they all drop their herbicide rate using fulvic acid rather than cultivating. But it's up to you, right? It's up to you. But cultivation is Armageddon. No, I didn't say that.
Yes, hello.
Yes.
If I would like to take in some amino acids or fulvic acids for human health, are there certain products for human consumption, or can I just drink out of the bottle by filling the sprayer, or?
Please don't do that. I use human grade, pharmaceutical grade fulvic acid. There's a lot of products on the market. You don't have to spend a fortune. They're not expensive. So there's a lot of these things are commonly used in health, which is the interesting thing about the connection between soil health and human health is the interrelationships, right? So use human grade, don't drink off the floor in the tractor stand thing. Don't do that.
Am I next? Okay. Thank you very much. Great talk. Thinking about fulvates and humates and their sources, can you get them from vermicompost, or what percentage come from sort of biological sources?
So I used to make a certified organic fulvic and humic acid from vermicast. So when you mix up vermicast and agitate it, the very dark brown substance is the humic and the golden color is the fulvic, right? But it takes a lot to make it, you're only making a little bit of humic when you're using like a 200-liter drum, right? So it's very concentrated. You guys, I think, have a lot of humic from peat. That's my least favorite. All right, I want ancient. Now it's a waste product of lignite, right? They've put it to the side. It's not a hot coal. It's a soft brown coal. It's concentrated. Secondary metabolites. Yeah. Thanks.
Hi. Read your book. I think it's great. And the podcast you did with John Kemp was great as well. I recommend that to everyone here. I'm making some compost extracts and have tried doing these aerated teas and find that it tends to select for flagellates or protozoa, and wondering what the benefit of this the boom bust cycle is in the did you say it was a 6-day 6-week aeration, and feeding it with the trace elements? Can you go into the benefits of doing that?
Because we're breeding up massive populations and we just want their metabolites, then they die, right? Some of them die, and then another organism's going to spike, and then it dies. So we're just concentrating the metabolites. I used to do commercial compost teas, and I don't anymore because you breed very narrow organisms in that compost tea process. Most people are spraying diseases on their properties. Stop doing it. You've got to be able to use a microscope to use good compost tea. So I'm not an advocate for compost teas.
Hi, Nicole. Couple of things. I find it ironic that Balance still sponsors your soil awards in New Zealand.
It is ironic, yeah.
And my other point is that I've heard you talk about native seed bank source, that quorum sensing sort of turning on those genes, and we've seen it at home in a very short time, and it just amazes me how quickly it happens.
And we'll talk about it in the next session, but it's phenomenal when you see it happening. You've got a seed bank there that's latent, and it's not going to change until you do something different. And when you see it come alive, like, it's mind-boggling. Love it. Is that it? Okay. All right. And so how long's the break?
30 minutes.
So I'll hopefully see you back here in 30 minutes. There's going to be no one here, and I'm like, okay, that sucked.